Alrighty. Well, hi everyone. Thank you for joining today. I will be presenting on a recent study led by Dr. Stuart Turvey and Charisse Peterson out of our lab at BC Children's Hospital in Vancouver, BC. This paper was recently published in Nature Communications just this past August, and I'm excited to share our findings with you today. Our lab at BC Children's focuses upon harnessing the early life gut microbiome to predict, prevent, and understand allergy. This is an important field of study as allergic diseases now affect about 20% of the population worldwide. Now, connecting this to the gut microbiome, the microbes living within the human gastrointestinal tract have been linked to the development of allergies, but there is still a bit of ambiguity surrounding how these microbes are exactly affecting the immune system and subsequent allergy. Specifically within this study, we used multiple allergic diseases as our outcomes of interest. These included atopic dermatitis, asthma, allergic rhinitis, and food allergy. We decided to look at all these diseases together due to the fact that they potentially have common immune mechanisms. These are typified by characteristics like type 2 inflammation and elevated IgE. Furthermore, they commonly manifest in a progressive or sometimes collective manner known as the atopic march. So therefore, studying them together, we thought might provide a better understanding of how they're both unique and similar in order to lead to improved preventive strategies. We were able to ask this question using the child cohort, and this is a longitudinal birth cohort in Canada that has followed over 3,000 children and families over the course of the children's growth and development from birth. The primary allergic outcomes we focus on here are allergic disorders as diagnosed by physicians or clinicians at 5 years. And we also utilized evaluations collected at 1 and 3 years, in addition to general early life characteristics, to really identify important early life factors in the development of pediatric allergic disease. So before diving into our microbiome results, we first compared the demographics and early life exposures of 592 participants with allergic diseases, be it atopic dermatitis, asthma, food allergy, or allergic rhinitis, to 523 control participants with no allergic symptoms across all the times they were evaluated by child study clinicians. So these are really robust controls that we use to compare the allergic participants to. When comparing the allergy groups to our healthy control groups, we found specific factors that were consistently associated with an increased risk of allergy. This heat map simply depicts the odds ratio using shading and indicates statistical significance of p of less than 0.05 using stars. So we see a particularly impactful effect by antibiotics, being biologically male, and being non-white. Next, moving on to our microbiome results on the whole, we analyzed both shotgun metagenomic sequencing data and metabolomics data collected from 3-month and 1-year participant stool samples to investigate the microbiome features and how they might be impacting the development of the immune system and later allergic symptoms. So these data sets allow us to analyze the microbes within the early life infant gut, encompassed within the question of who's there, as well as their functional potential and their associated metabolites, encompassed within the question of what are they doing. So to start, we first looked at the microbiota present within the infant gut. It's important to note that changes in the early life gut microbiome occur in concert with immune development, and the microbial components within the gut can affect immune programming. This is exemplified in things like short-chain fatty acids, inducing differentiation of Treg cells, indolactic acid preventing the expansion of Th2 and Th17 cells, and so on. So the appropriate progression and fluctuation of bacterial populations is therefore essential to proper immune development. Given this importance of microbial population progressing in a proper way in order to facilitate proper immune regulation, we explored microbiota maturation in the infant gut. We did this through the creation of a microbiota predicted age variable. We did this using random forest, and found that the structure of the microbial species within the infant gut was so linked to infant development that it could be used to predict participant chronological age at the time of stool sample collection. Here we see the scatter plot of the microbiota predicted age on the x-axis and chronological age on the y-axis. So this allowed us to compare the development across our groups and identify any irregular patterns that may have been developing in allergic participants. Indeed, so using this predicted age, we were further able to discover that participants with allergic diagnoses at 5 years had a consistently lower predicted age at 1 year, despite having nearly identical chronological ages. And the means of each metric are shown by these either vertical lines for the microbiota predicted age or horizontal lines for the chronological age. Importantly, this remained consistent across all the individual diagnoses as well. So we next looked at the functional potential of the microbiota present. Using the Metapsych database, we identified pathways that were both significantly associated with predicted age, as well as associated with having two or more clinically distinct allergic diseases at 5 years. Using these uh, Using these criteria, this resulted in the identification of 11 pathways meeting our significant thresholds. Overall, we observed two distinct trends within this analysis. The first being an increase in the potential for inducing an increased oxidative state and mucosal breakdown within participants who would be later diagnosed with allergic diseases. And the second trend we saw was that there was a decreased potential for secondary fermentation, short-chain fatty acid production, and mucosal strengthening activity within these same infants. Finally, we analyzed the metabolites within the infant gut to gain additional insight into the biological impact of the microbes and to identify targetable biomarkers of allergy risk. An important aspect of this analysis was a simple Spearman correlation analysis between the pathways identified in the analysis I outlined previously, in combination with important microbiota in our predicted age model, seen in this heat map with metabolites on the horizontal axis and microbial features on the vertical. The primary findings from this investigation included identified a significant association between pathways associated with an increased risk of allergy and a low microbiota predicted age and these trace amines. Now, these metabolites
Courtney,
are fairly,
Courtney,
yes.
sorry. We actually don't see the slide changing anymore. We are stuck at slide number
Oh,
10. Maybe
oh, you're
like,
slide number 10.
yes,
Oh, that's
sorry.
quite far back. No, that's okay.
Um,
Let's see. I will stop sharing and try
yes,
again.
let's try to reboot that. Thanks.
Oh boy, I think I was on slide 19.
Okay, well, we did what you were saying. It's just that it took us a while to figure it out that the slides
Mm,
were supposed to change. And
yes, okay. That's good to know. Alrighty, can you see slide 19?
yes, we can. You
Okay.
can go back a couple of slides if you want to.
Mm-hmm.
Okay.
That, do we have time for that, or do you want
Um,
me to just
we-
continue from where I'm at?
I think we have time. Yes, don't worry.
Okay. Okay. Let me know. So you said slide 10?
Yes, that's correct. If you can just briefly like show us what you were talking to us about. Thank you.
Okay, yeah, of course. So not sure where it stopped, but here we're using a predicted age to discover that participants with allergic diagnoses at 5 years had a consistently lower predicted age at 1 year, despite them having nearly identical chronological ages. And we saw this across all the individual diagnoses as well. And please let me know if you can still see or if it stops changing for any reason. But Our next analysis looked at the abundance of functional genes in order to look at the functional potential of the microbes that we were seeing within the gut. So looking at the functional potential, we used the Metacyc database to characterize the different sequenced pathways and genes that were associated with these microbiota. And we are able to identify 11 different pathways that were both associated with our predicted age metric as well as allergic disease. And within these pathways, we observed two important trends. The first being that there was an increased potential for inducing increased oxidative state and mucosal breakdown within participants who would be later diagnosed with allergic disease. And the second trend was that these same participants had a decreased potential for secondary fermentation, short-chain fatty acid production, and mucosal strengthening um, within the participants who would later develop allergic disease. And then our final analysis, we looked at metabolite concentrations using NMR and LC-MS within the gut, and we analyzed these metabolites to look at the broader biological impacts that the functional potential and the function of the microbiota might be having upon the infant during their early life. And here we see the Spearman correlation heat map that I was talking about before. So this was a really important aspect to look at what metabolites were being associated with those important pathways that were associated with the predicted age as well as the allergic diseases. And the primary finding from this analysis was this high association between the pathways, important microbiota, and these trace amines within the infant gut. Now, trace amines are fairly underexplored, but they have been seen to influence Th2 responses and increase inflammation within the gut, and this would potentially exacerbate an allergic-prone immune system in the early life. So finally, this is an important closing investigation from our study that involved using causal mediation analysis through structural equation modeling to see whether the associations that we identified could rise to the question of causality. And we did, in fact, identify that important pathways and metabolites, as outlined here, collectively within a latent variable had a significant indirect effect, meaning that this relationship between microbiota predicted age and having allergic diseases or diagnoses at 5 years was or could be potentially be caused by these altered components and metabolic profiles of the early life gut. So overall, thank you for listening to that again. But pathways and metabolites involved in increased oxidation, mucosal breakdown, and Th2 skewing show trends of increasing the risk of allergy stemming from a lower microbiota maturation metric, while pathways and metabolites involved in fermentation, tolerance reduction, and mucosal strengthening show trends of protecting against allergy via an increased microbiota age. Again, thank you all for listening, and that's all from me.